My hope today was to talk about a hypothesis about myocarditis and why it is being triggered by mRNA vaccines of the type we have seen. Now, I'm not going to present this as factual. I do not know that this is true. But what I do know is that an understanding of a bit of biology, an understanding of what viruses are, about the predicament that viruses put multicellular creatures like us in, and the way that our ancestors have evolutionarily learned to fend off viruses, suggests a hazard about this new and amazing, but until now untested technology when introduced into the human body. So that's my hope. And I think the thing to do here is you're going to play student. Okay. And I don't mean student in the sense that most people are familiar with it. I mean it in the sense that we scientists become each other's students at the point that we have something to discuss where we don't know, right? We are the students of those who have seen something we have not yet seen. So you're going to play that role on behalf of the audience.
Which means explicitly the expectation is that I interrupt you when you say something that I don't find clear or that I maybe disagree with.
Yeah, maybe disagree with, and maybe I learned something because you forced me to see something about what I'm saying that I didn't see when I went through it myself. Something like that. Okay. And, of course, the lovely thing is that this is exactly how we behaved in a classroom. And we had a group of, if we taught together something like 50 students, but we would also be each other's students in that context. And certainly we have different areas of specialty, and it all works out pretty well because really what you always want when you're trying to understand something is somebody who understands why you don't get something yet to ask the question that causes it to be explained in a way that you do get.
Well, and potentially model what other people may or may not already know. Right. That's so the difference between, say, the dinner table and a classroom is that the classroom is going to be a representative sample of at least, some larger group. And you will hear from people, if you hear from a few people, that isn't making any sense to me. There's a decent chance that either there's a lack of understanding in the population that you didn't know was there, or there's a way that you're communicating it that is in some number of people's blind spots. So let's try this. This is tougher than either a dining room table or dinner table or a classroom, because the audience really is a lot of, it's all of you who we can't hear.
We can't hear, and we don't know in detail, so we can't even guess what is and isn't clear.
I mean, we hear a little bit, and in fact, we'll end maybe today with one of so many lovely notes that we got in these last couple of weeks from people, but we can't hear in real time. So anyway,
right. And I would just say that there are many, many different moves that the student in this case is entitled to avail themselves of, including, here's a question. I don't think the audience will know X piece. I think I know it, but I'm going to ask you the question anyway to get you to spell it out. So anyway, you have tools.
I don't know if what you just did was rhetorical, if you're about to now ask me something.
Nope.
Okay.
That was rhetorical. Okay. That was just pointing out that I am not necessarily, in explaining this to you, I am really using you as a surrogate for people, right, most of whom aren't biologists. And so anyway, I'm going to err in the direction of being overly clear, hopefully. All right. So here's the predicament. Viruses, I believe I'm going to oversimplify everything because that's what we do in science, especially anything in the area of complex systems. The fact is we do not fully understand biology. We're a long way away. So what we do when we discuss biology is we describe a cartoonish version of the reality. And hopefully our cartoon is simplified but not incorrect, right? We describe an overly simple version of the reality. And hopefully if you were to expand it to the full level of complexity, it wouldn't contradict what you say. That's the objective. Mm-hmm. So I'm going to describe enough about viruses and enough about the immune system for you to see a picture that I think I increasingly see clearly, right? That may explain why myocarditis is a downstream effect of RNA vaccinations, or at least some of them. And in so saying, I do not mean that this is the only reason right? We have different kinds of hypotheses. You've got mutually exclusive hypotheses where if this is causing it, then that isn't causing it. But in this case, I'm just simply saying here is one mechanism that I believe is likely to create a link between these things, and there may be others.
Okay, let me ask you right up off the bat then. You are going to propose a hypothesis that posits an explanation for why myocarditis is downstream of these mRNA vaccinations. Of course, we now know that myocarditis is one of the sometimes effects of SARS-CoV-2 infection, of the actual disease COVID. Is this a separate hypothesis from that? And if so, does this explain that separately? And if so, do you have a separate explanation for why you think SARS-CoV-2 may also be causing myocarditis?
So myocarditis, my understanding of it, and I'm not a heart specialist by any means, sure. My understanding of it is that it is effectively inflammation of the heart muscle, and inflammation can be downstream of various other processes. Now, my guess, and I do not know, but my guess is that the inflammation caused by COVID is different in origin than the inflammation caused by the mRNA vaccines. And I believe I have learned this by listening to Dr. McCullough, who mm-hmm. is a cardiac specialist. Mm-hmm. And I should also point out, as I was formulating my hypothesis for what's going on here, I shared it with several people. And one of those people, Alexandros Marinos, said, I believe I have heard that hypothesis elsewhere. And he, in fact, pointed me to a place where Dr. McCullough appears to spell it out. Mm-hmm. And I also now know that our friend Jonathan Cooey, who some of you will know from his lab leak series early in the pandemic called JC on a Bike, Journal Club on a Bike, that he also has advanced a version of this hypothesis. So I don't claim it to be unique. I do think I arrived at it independently. Mm-hmm. But in any case, here's the basics. We multicellular creatures have a problem with pathogens, and pathogens come in different flavors, right? You've got things like fungi, a comparatively rare pathogen for us, bacteria, a comparatively common pathogen for us, and viruses, a very common pathogen. Now, you want to compare in your mind the difference between bacteria and viral infections. Bacteria are living creatures, and as such, they have a biology of their own. It is simplified relative to us eukaryotes, right? Because bacteria are smaller and simpler than we are, but nonetheless, they are biological creatures in their own right. Viruses are an interesting kind of exception. They function biologically, but they are definitionally in a gray area. And the reason for that is that viruses have abandoned the mechanisms that allow a biological creature to function. Now, how is it that they function if they've abandoned those mechanisms? Well, they borrow those mechanisms. So they are in some sense analogous to special forces that you might drop behind enemy lines that would not carry a great deal of equipment with them, but would have the knowledge of how to utilize stuff that they find inside of enemy territory for their own use. So instead of, getting tanks and airplanes and things in behind enemy lines, you drop something that is full of information about how to utilize what they find locally to the same effect. And viruses are effectively this. They have gotten a tremendous advantage by abandoning their own biology and commandeering the biology of the creatures that they infect once they invade, right? And what this means is that we have a problem in trying to fight them, because almost everything that viruses do isn't really them. It's our own machinery that has been commandeered and has now been turned against us.
Okay, can I interject here?
Please.
Just a point of clarification, which I think is not at all central to your argument, but I believe, and I think I may have missed something, but I believe that you said pathogens can come in the form of sort of fungus and bacteria and viruses. And it seems to me that fungus is a really tiny tiny kind of human pathogen, but there's 2 large classes that aren't included in that. So just in service of sort of categorizing what kinds of human pathogens there are, of course, single-celled eukaryotic pathogens like Plasmodium, which is the microbe that causes malaria.
Yep.
So, bacteria are single-celled prokaryotes without a cell nucleus. And then there are single-celled eukaryotes, what used to be called protists, I think, but it's sort of a giant junk group. It doesn't have a single evolutionary origin. And then, of course, there are multicellular organisms, animals like worms.
Worms, sure.
Right. So just to have like a categorization, there, pathogens come in all of these, single-celled, multicelled, all of these things. The distinction really that I think you're trying to make here is virus versus not virus, right? Where, the virus, and, there are lots of other functional ways of discriminating between them, but the particular distinction that you're making is because a virus has stripped itself of almost all biological functioning, it can act as effectively special forces.
Yep.
Right. It can come in, and you didn't say this yet, but you've said this to me before. It can come in sort of like the dude with the backpack and sneak in without having all of, it doesn't have a tank, and it doesn't have, all the stuff that makes it easy for the body in this case to say, there it is. It's easy for it to sneak in under the wire and without alarming the self-defense.
Okay, so I want to add 2 things. One, there's even a weirder category than virus we now know. This is something that was very much learned during our lifetimes, which is that there's also something that's even more stripped down, which is a prion disease. And it's not even really clear that a prion disease, it's not clear that it's serving its own interests. A prion disease is a contagious misfolding of proteins, right? And so anyway, it's very pathogenic from the point of view of doing harm, spongiform encephalopathy being, mad cow disease being a classic example of this. But, a virus is at least creature-like in the sense of we know what it's trying to accomplish. A prion disease could just be an error that echoes, right? I'm not going to pretend to know. I'm sure there are people who know better than I do, but nonetheless, the spectrum goes from things like a worm, right, which is similar to us, a multicellular creature, to single-celled creatures, to non-celled creature-like things, viruses, to folding errors, right?
Yeah.
And all of these things have pathological implications.
Yep.
But let's take your example of the special forces guy who parachutes behind enemy lines. Let's say he gets a tank. He gets behind enemy lines, he sneaks up behind somebody who's got a tank, slits his throat, and takes over the tank, right? Now you've got a question, which is your own forces are now looking at a tank that looks like them. That's our tank, but it's pointing at us, right? So the point is, which tanks of ours do we now shoot at, right? And that is the question that viruses create. And so without going too deeply into the issues of immunity, the overarching story of immunity for creatures like us is something called self versus non-self recognition. What happens is, early in development, there is a process in which your immune system, which in principle is capable of responding to effectively any biological molecule, that immune system is asked a question before you encounter the outside world. And the question is, which things can you currently see? Now, if during that period, in utero, you see molecules that are biological, well then, okay, those are yours, right? They almost have to be, right? Because you're insulated from the outside world. And so,
well, they're yours, they're mom's.
Well, but that's just the thing. Even that, the placenta is a absolutely remarkable organ that does not get enough attention, right? The way in which it allows mother and baby's systems to interact without exchanging very much above a certain size is miraculous. But the point is, the self-non-self recognition system is programmed during development to recognize all of the stuff that you yourself produce, and then to go on for the rest of your life and ignore it, right? Then, once you build a system that recognizes all of the molecules you produce and ignores them, now it can do something else, which is now anytime it sees anything that isn't on the ignore list, it can assume it's a pathogen, right? That means that you don't need to have seen a particular pathogen before in order to know to fight it.
Right.
Right? So
it's a simple rubric that can be gamed.
And also has certain failure modes, like allergy.
Right.
So if pollen gets into your lungs, well, your immune system won't recognize it as self, but it's not a pathogen. And so fighting it can be worse than ignoring it, but you don't know that it's actually should be on the ignore list. And there's lots of interesting stuff about the IgE system. Ig is the abbreviation for antibodies, which are the most famous component of this system, which we know, which has also allowed our minds to be gamed. But nonetheless, IgE is one of the subsets of antibodies that appears to be particularly prone to create allergic reactions. And it's probably particularly prone to create allergic reactions because it's not busy doing the things that it would normally do, like fight worms, which we don't have a problem with in the first world.
Because we have such a sterile landscape that we've created for our bodies and our environments.
Right. So you get some part of your immune system that's twiddling its thumbs and it overreacts to,
Yeah.
It's the bank guard.
Grass pollen. It's grass pollen.
Right.
Like, could you just sit down? just relax.
We got this. It's grass pollen. But okay, so you've got a system, and it functions basically by ignoring everything that you make and therefore feeling entitled to react to anything that you don't make. Now, again, remember what viruses are. Viruses are these, they are like particles with a creature-like objective, but no creature-like bits, right? It's just like an envelope with creature-like objective written on the letter inside. And that letter, if it gets into your cells, instructs the cells to do what the virus, if it were a creature, would do for itself, right? Make more viruses. So here's the thing. When viruses get into us, wherever that happens, if they're respiratory viruses, it will typically be in the surface of the lungs, for example. We get other kinds of viruses, it will be in other tissues. But the virus gets in, if it manages to get into a cell enough that its message can trigger the cell to start doing the virus's bidding rather than the creature's bidding, then that cell becomes effectively a factory that is dedicated to the virus's interests. Now, the body has to now look at this the same way you would look at a tank that had been commandeered by enemy forces that had parachuted over your border and grabbed a tank. Right? It has to say, aha, I used to think that was self, but that one drives different, right? That one drives on the left side of the road, not the right side of the road or something. I got to shoot at that one as if it's the enemy because it actually is.
Even though it also has the tags that say self.
Right. It has the tags that say self, but it has indicators that it is not anymore self. And so the basic punchline here is that when our body sees a cell that looks like us, but does something extra. In other words, it puts out messages that look like non-self. It concludes that is a virally infected cell. And the right thing to do is then to kill it, because a virally infected cell is not going to become unvirally infected. So there's no trade-off, like how much is that cell worth to me, dead versus alive. The point is, once it's been commandeered, even though losing a cell is not a good thing, it's better than that cell continuing to do the enemy's bidding. So the immune system targets cells. And in fact, the cells themselves have an interest in being targeted because they only pass on their genes if the creature survives, and the creature survives better if it's not infected. So the cell effectively would volunteer for death. Okay. But the basic point is, what does a virally infected cell look like? It looks like a weird hybrid between self and non-self, right? It has self markers because it's a self cell, and it has foreign markers because its internal machinery has been commandeered and it's now making proteins that the body has never seen before, et cetera, et cetera. Okay, so now what happens if you do what these mRNA vaccines do, right? If you just assume that they function as the brochure tells you that they should. These vaccines contain a message. They are viral-like in this way. They contain a message without biological structure around it. And it is encoded, or it is
It's even more stripped down than the virus itself.
Yeah. It is a single message in this case. These are cartoonishly simple vaccines in the sense that they produce one antigen, the spike protein. And that spike protein is wrapped in lipid nanoparticles. Now, why is it lipid nanoparticles? It's lipid nanoparticles because like dissolves like, and by surrounding these things in lipids, they are protected from the parts of the human system that would disassemble.
All the mRNAs, I'm not sure how to pronounce that, but that are in the intracellular space would immediately tear apart the mRNA if it weren't protected.
Right. So by putting it in this envelope, they make it invisible to a system that would otherwise attack it as foreign. And they also cause it to have an affinity for human cells. Those human cells, all cells are covered in this lipid envelope. Right. And so because like dissolves like, the lipid, and this is one of these places where the story we tell isn't exactly right, the lipids, being nonpolar, are not actually attracted to each other, but they're driven together by all the polar stuff sticking to itself. So,
well, and the lipids on the cell is actually this phospholipid bilayer, which has things sticking inside, which, anyway.
Yeah. Anyway, so the point is the lipid coat, the lipid nanoparticle coat, causes the mRNA to get taken up by cells. And those cells, because of the way the cells are constructed, so the cells are constructed, they have a nucleus that has...
They're normal human cells.
Normal human cells. They have a nucleus with a DNA genome in it. Certain mRNAs are produced from genes that the cell wants to transcribe. And... they're produced from genes that the cell wants to transcribe. Those mRNA messages move from inside the nucleus into the cytoplasm. And in the cytoplasm, you have ribosomes that automatically translate mRNAs that they encounter. So the whole clever thing about these vaccines, and it is truly clever, is if you put an mRNA message into the cytoplasm of a cell, the cell automatically transcribes it because the cell is built to do that, because it wouldn't encounter mRNAs in its cytoplasm other than in the case of maybe being infected.
It's the factory behind enemy lines. If the virus is the enemy and the virus is dropping in, you're like, okay, can we get self to make things that look like you to trigger the immune system to say, aha, now I know. Yeah. And once informed, I will not forget.
Right. Okay. So you get cells. They pick up the mRNA from the vaccine. They start producing spike protein. Now, the design of these vaccines, right? The spike protein has been modified. The hope of that modification, which failed, actually, so it doesn't completely do this, but presumably it does it somewhat.
Modified from the so-called wild-type SARS-CoV-2.
Right.
Yeah.
So it's been modified in a couple ways. It's got a hinged piece that's been locked open in order to expose a piece of the antigen to the immune system that would otherwise be, because the virus is a very sneaky evolved creature, it's covered in sugars basically that shield what would ordinarily trigger the immune system from seeing the spike protein. So they've locked it open so the immune system sees it. But anyway, the point is they put in this anchor domain to get these spike proteins to stick out of the surface of the cells, the hope being that the immune system will see these antigens sticking off the surface of the cells, will learn what they look like, and you will magically become immune to the pathogen when it shows up with very similar antigens on its surface.
Immunomagically.
Immunomagically. Well said. Okay, here's the problem. A cell that is displaying the spike protein on its surface will look, in all probability to the immune system, like an indication that that cell has been virally infected, because that cell is now displaying a mixture of self and non-self proteins.
Yep.
And the immune system has no model for this, because the only time that would likely take place under natural circumstance is viral infection. The right response to a cell that is doing that is to kill it, because as much as that cell is useful, it has been lost, right? If your enemy has captured a tank of yours, you're better off, even though that was your tank, you're better off destroying it, because at the moment it's working for the enemy and not you. So the immune system, seeing cells that have taken up the lipid nanoparticles and transcribed the message inside, the right response for the immune system is that is a virally infected cell and it has to go, right? Now, this is the wrong thing. If you could tell the body what was really going on, what you would want to say is, look, stand down the immune system, because that cell that looks like it has been permanently compromised has only been temporarily compromised. Those mRNA messages will eventually be lost and the cell will go back to doing its own bidding, which is not something that happens in a virally infected cell. So the immune system reacts, it overreacts, right? And it destroys these cells. Now the problem is, and this is where this whole set of questions that we've been dealing with over the course of COVID interacts with all the weird stuff I did as a graduate student surrounding telomeres and cancer and tumors. The heart is a weird tissue. Now, the reason that the heart is a weird tissue is that the heart does not have normal repair capacity. Almost all of your tissues have the capacity to replace dead cells with new cells and to keep functioning. It always comes at a cost because you're always borrowing from the future capacity to repair that tissue and maintain it. So every time you destroy some tissue and have to replace it, you're effectively accelerating your aging. But the heart is super special. It's one of a small number of tissues that doesn't do that trick. And what that means is that when you destroy a cell in the heart, it doesn't get replaced by a similar cell, right?
This is maybe too far afield, but it strikes me that the heart not having repair capacity is for analogous reasons to salmon being semelparous. That it is about the chances of surviving repair, or in the case of salmon, the chances of surviving another trip back out to the ocean and years and another move back into the freshwater where you came from and having another reproductive opportunity. The chances in both of those cases are so low that in the case of the heart, the repair capacity has been dialed all the way down. And in the case of salmon, they've become a, breed once then die species.
Well, interesting. I think there's an analogy. I think it is actually different, but if I can
It's a different analogy.
Yeah.
Okay.
If I can rephrase what you've said,
Yeah.
there's a point at which your likelihood of making it to a second reproductive event is so low.
If you're a salmon.
If you are a salmon, or really if you're anything. There's a point at which if you are at reproduction, your chances of getting back to reproduction are so low that you ought to invest everything in that reproductive event that you've reached. And therefore, there's a shift where it's like anything you hold back is wasted in all probability. So you might as well invest it all here. That is to say, any tissue that you can turn into more eggs or more sperm is better spent now because it's very unlikely to get back to this very difficult place.
Yeah. Specifically Pacific salmon we're talking about, not Corinchus.
Right. And so anyway, there's a whole interesting discussion here. I have hypothesized that actually bears may be the difference. It could also be the sharpness of the Western mountains. But the point is the stakes are very different in the West part of North America than the East in a way that has caused all but one of our Western salmon to go semelparous.
Or salmonids, yep.
Yeah. Okay. So back to the heart. Why is the heart a different case than the salmon case?
Yeah.
The hypothesis I put forward in my paper with Debbie Cizik I guess it was published in 2002 finally.
That's right.
The hypothesis that we put forward was the reason that the heart lacks this capacity is that in order to have the capacity to do cellular repair, you run the risk of creating what we called a prototumor. A prototumor is a growth of cells. If a cell becomes unregulated,
oh yes,
and it produces a bunch of, if a cell becomes deaf to the messages that say stop dividing, then it will just simply divide. And if a cell does that in the heart, it could create a prototumor. Let's imagine something the size of a pea. Well, a growth the size of a pea, if it happens in your liver, you're never going to know about it. If it happens on your skin, it will be a mole. If it happens in your heart, it could block an artery and kill you, right? So the point is, the danger of a prototumor is so high. And again, this is a hypothesis. I don't know if it's true. And as far as I know, it's never been tested. But the danger of a growth of cells out of place in the heart is so large that the system has had repair turned all the way down so that there is no hazard of that. And as a result, the heart is this absolute marvel of design, or as if design, where it goes for 85, 100 years effectively, flawlessly, without the capacity for repair. And there will be people saying there is capacity for repair. Yeah, there may be a tiny bit. In general, though, instead of repair, what you get is the capacity to scar, right? And scar is basically like biology's spackle. Right? It's not the thing that is being replaced. It's something that's good enough. And so in any case, to make a long story shorter here, you've got an mRNA message which causes cells to transcribe or to translate a protein that is not self, which almost cannot help but be read by the immune system as an indication that those cells have been virally infected, which they haven't been, but the immune system doesn't know about mRNA vaccines, and so it will assume that the category that explains this anomaly is infected cell, and it will do what the right thing to do is, which is to destroy that cell, when in fact the right thing to do here is to leave that cell alone, let the mRNA messages burn themselves out, and let the cell go back to its job. Because damaging the heart is not a good idea.
Right. Damaging the heart is not a good idea. I think one of the pieces I see missing yet is, obviously it was hoped, it was expected, we were told that the mRNA vaccines would stay basically local to the spot of injection. And it is well documented at this point that that has not happened, is the idea that they spread, and maybe there are predictable, maybe we could make predictions with regard to, whether or not the proper aspiration happened, whether or not it was left or right arm, a number of these things have been proposed, where it will spread to, or is the prediction that the vaccine is going to be spreading throughout the body, much to the chagrin of those who developed the vaccines, and that doesn't have necessarily widespread horrible effects except when it ends up in the heart. It's not that it is specifically going to the heart, it's that it's ending up everywhere, which it never was intended to do. And when it ends up at the heart, that's going to have particularly bad effects.
Yeah, if the hypothesis is correct, it is a close analogy for what I found to be likely with respect to drug toxicity back in the days of my telomere work, which is we see the heart damage for a couple of reasons. One, because the heart is very bad at self-repair, so damage shows up in a meaningful way. The same amount of damage in most other tissues would never bother us, right? You'd get better and you wouldn't know that you'd lost some future capacity for repair. And B, the failure of the heart is utterly unambiguous.
Right.
So let's say that the vaccine robs you of a bunch of liver capacity. Well, good news. You were born with excess liver capacity. And so the point is probably you don't die of liver failure, you die of something else, so probably you got away with it. Right? If it's the heart, well, you probably didn't get away with it. You've borrowed from something important, right? Heart failure is one of the major. So you've got basically two important biotic ways to die for a well-fed, well-protected creature. One of them has to do with growth of tissues that shouldn't grow, basically tumors and cancers. And the other has to do with the failure of organs that are necessary. And heart being primary among the organs that are likely to fail and kill you. And the point is, when this happens in somebody young, it's conspicuous because it doesn't happen in somebody young under ordinary circumstances. So when somebody drops dead on the football pitch, that catches our attention. Now, it's not that that never happens under normal circumstances, but there's a question about, is it happening more than we're used to, and is there a reason for that? And basic point here is that you've got a mechanism of action, the expected mechanism of action, the desired mechanism of action is for this message to be taken up by cells and translated into protein. That is likely to trigger the biological phenomenon of non-self recognition and the de facto conclusion of the immune system, this is an infected cell and it needs to be killed. When that happens in the heart, it is both worse for you because your heart's not good at dealing with it, and more conspicuous to medical science because somebody who shouldn't have had a heart attack just did.
Right.
And that is resulting in this. Now, you raise an interesting point about the potential asymmetry. And this is one of the little corollary hypotheses here, which is that if this is true, if this is the mechanism or one of the mechanisms that is causing serious myocarditis to come to people who have been vaccinated with these mRNA vaccines, there's a question about whether or not, what is it that causes it to be taken up by the heart, right? The thing that is lacking in these vaccines, right? If these vaccines were really to be brilliant enough to be wielded in this way, they might have a targeting mechanism that would cause them to be taken up only by tissues that you could afford to suffer this fate, right? And not to be taken up by tissues that you very definitely wanted to protect from it, like the heart. And they don't have that. They've got no targeting mechanism in them at all. So what happens is you have an arbitrary uptake, right? It's not random because some cells are more prone to take up the mRNA, the lipid nanoparticles, than others. And the heart, as I understand it, is prone to take them up. So if they flow to the heart first, if they escape the arm and they flow to the heart, then that's a very likely place for them to be taken up. And then what you'll end up with is dead tissue in the heart that then hopefully scars over, and hopefully you close to get away with it, but you're losing capacity from your heart for life. But there's all kinds of questions. If this is actually true, and again, this is just a hypothesis, but if it's actually true, then it raises all kinds of questions about how might we have vaccinated people that would reduce this effect, right? So for example, should you be getting this one in the arm? Would you be better off getting this shot in the ass, right? It's possible that the circulatory pattern would be less devastating because the stuff would get taken up before it gets to your heart. Now, I don't know enough about circulation to say maybe it would make things worse if they gave it to you in the butt cheek. But that's one possibility. Another possibility is that there's asymmetry in the circulatory system, right? Heart attacks cause left arm pain. And so there's a question. Yep. We don't know because we didn't get this vaccine, but it's possible that they sit you down, they say, which arm would you like this in? And it's possible that shouldn't be your choice, that they should give it to you in the arm that protects your heart better, right? So anyway, there are all kinds of questions you could ask here. And because what we've been fed is, these things are safe, we can't, John Campbell did a marvelous exploration of the question of aspiration, right? It's possible that these vaccines are simply not safe enough to do this, but they could be rendered a lot safer than they are by things like aspiration technique, by tissue in which they are injected, and that we aren't asking those questions because instead we've been fed a fairy tale about safety.
Right. Myocarditis. The fact that only some people suffer post-vax, is it due to random distribution of spike protein, bad luck, or might some people be more susceptible to due to genetics, or some tolerate it better and never notice the weaker course of illness?
Sweet.
Yeah.
We can do this.
Yeah.
So,
it's written sort of telegraphically, so I was just trying to translate it as I was reading it.
Many things are true.
Yep.
One, there is liable to be a roll of the dice thing, right? Like you've got an actual volume of liquid with actual stuff in it, and it is going to circulate in some chaotic way. And to the extent that particular lipid nanoparticle coated pieces of RNA bump into the walls in the heart and then get absorbed, whereas others were, 3 millimeters away from the heart as they went through to tissues downstream. All of that chaotic flow stuff is going to mean that some people are going to get a huge dose in the heart, and some people are going to get a very low dose in the heart. And the aspiration thing that John Campbell surfaced may have everything to say about this, because the point is maybe everybody who gets a huge dose in the heart are people where the needle actually hit a circulatory vessel rather than the interstitial space.
There might be something about the variation between batches, although I don't know if we know anything about what the variation between vaccine batches, actually means in terms of how it manifests in the body.
Yeah, if it's even a thing, and it's tough to chase down. There could be heritable differences? My guess is it's very small.
Yeah, me too.
If it's a component at all. I think
because it's your heart, right? Because you simply, there's simply no backup, and if it gets there, it's going to be a problem for you. I was thinking actually when we were talking in the first hour, what else? Well, and obviously you and Debbie spent a long time thinking about this for, many years, but, what else might be like the heart and therefore you would predict to have really low repair capacity? And the one thing I came up with, and it's not perfect because we got two of them, is a loop of Henle in the kidney, not kidney tissue writ large, but specifically the mammalian synapomorphy, the unique character of mammalian kidneys that allows for, basically the recovery of more water and the excretion of liquid waste through that thing that's called the loop of Henle, which is so thin that, you know, a problem there could similarly take one of your kidneys offline. Potentially right away.
You've got so many actual loops of Henle, right? I mean, these are tiny structures, and you have presumably millions of them.
No, it's I hope I'm not wrong here, but you know, there's lots of little things off of it, but like in the main loop.
I'm going to stay out of it because I don't know.
Okay. I think you got 2 basically.
Well,
but maybe I'm wrong about that.
Let's see. You've got other tissues that behave in a weird way.
Mm-hmm.
And I would argue what I argued in the paper back in the late 90s, early 2000s was that certain tissues have reasons not to have high repair capacity, and that all of them are low cancer risk and high wearing out potential. That basically that's the pattern, that any tissue in which you've got high repair capacity has a substantial risk of tumor growth and a substantial capacity to repair damage, but that things like the eye has a low capacity for repair and tends to wear out. Cartilage has very little capacity for repair and tends to wear out.
And you don't get cartilage cancer, and you don't get eye cancer, but you get like optic nerve cancer. Like there's nervous tissue cancer, there's blood vessel cancer, but the eye itself doesn't tend to get cancer. Right. Is that right? Yeah.
Right. And the heart doesn't tend to get cancer.
Right.
The pericardium, the membrane around the heart, does when people get cancers in that neighborhood. That tends to be the pericardium. But anyway, it's a general
That's
pattern.
interesting because, of course, myocarditis and pericarditis are the two kinds of inflammation that are really happening here. And all of the discussion that we had earlier was about myocarditis, not pericarditis, which is an inflammation of the pericardium, which is the membrane around the heart.
Well, actually, it reminds me of something
the pericardial cavity.
I forgot to say in our main podcast, which is that effectively the hypothesis that I put forward is a hypothesis of temporary autoimmunity, right? The
Explore
idea
that.
So we've got autoimmunity is the immune system mistaking self for hostile and attacking self.
Mm-hmm.
My argument is the mRNA in heart tissue is likely to trigger the immune system to attack the heart, but the mRNA transcripts that cause the production of the spike protein are presumably transient. And so the point is, if the immune system were to hold back and not attack these cells, the cells would go back to being normal. Mm-hmm. And so the point is, it is a temporary autoimmunity that causes the permanent destruction of cells in the heart that then results in the heart being effectively enfeebled, damaged.
Mm-hmm.
So anyway, I wish I'd said that in the main podcast, but it is what it is.
That's what it is indeed. And then there's more categories than this, but this does seem like a good group of 3. Is it luck? Is it genetics? Is it heritable? Or do some tolerate it better and never notice the weaker course of illness?
So this is a biggie.
Yeah.
If the hypothesis is correct, one of the things that it predicts is that there will be a ton of subclinical myocarditis that will never be described.
Yeah.
And what that means, the way we will see it manifest, is shortened lifespans, right?
Yeah.
That a large fraction of people ultimately die from heart failure, and that those deaths will happen earlier than they would otherwise have happened because their heart has effectively expended what repair capacity it has early in life and been caused to be less capable. Now, this, of course, fits with the observation, which I believe is likely to be robust, that we have an unusual number of people dying during sporting events. And the basic point is that athletes are being pushed to a limit that most people are not regularly pushed to. And that the point is a heart that can function below that threshold will suddenly fail when stressed to a point because it has lost some of its heart capacity to vaccines that trigger this temporary autoimmunity.
Yeah, and there's something new out, which I haven't tracked yet, about catecholamines and, sort of having an environment in which adrenaline and noradrenaline and dopamine, I think is the third catecholamine, being at elevated levels may make heart failure in the already compromised heart more likely, the myocarditis in an already compromised heart more likely. But I can't track it more than that yet.
Yeah, it's on my list of places to delve, but I haven't delved into that result yet.
Can I have myocarditis without symptoms? Will it go away? I got tinnitus 3 days after the booster. Since I have no recourse, should I cheat on my taxes since the government cheated me?
Wow.
Yeah, you shouldn't.
Not cheat on your taxes.
You shouldn't, but they really are not holding up their end of the bargain, are they? Nope, they are not. They are not. Can you have myocarditis without symptoms? Yeah, I think this is the subclinical stuff that you were referring to, and for sure there will be people who have damage and who are never aware that they had damage, and may never become aware that they have damage, or may become aware later. Oh, it looks like you have some past damage. Your doctor tells you 20 years later, and you say, nope, I really don't. Well, could have been that injection.
Yep.
Anything else to say here? Will it go away? It does. It seems to pass, but it doesn't. It leaves scarring.
Well, it will reduce the capacity of your heart, presumably for life. Now, I want to take that back.
Yeah, I don't know that that's true.
No, I believe it's true.
Okay.
But let's put it to you this way. I think what I've learned from Peter McCullough is that you really have two kinds of myocarditis. You've got an inflammation that goes away, but is not the result of cellular destruction. That the myocarditis that is the result of cellular destruction will permanently reduce the capacity of your heart, which, depending upon what you do, if you're an elite athlete, probably you won't be an elite athlete after that happens.
And McCullough's argument is the kind of myocarditis that does not involve cellular destruction is the kind that SARS-CoV-2 causes, but that the kind of myocarditis that does cause cellular destruction is brought on by, presumably among other things, the mRNA vaccines. Is that the right categorization?
I think so. Okay. I don't want to represent his position because if I've got it wrong, I've got it wrong. But yes, I believe that is his argument.
Competing hypothesis. Blood goes to the heart twice before going on to liver, kidneys, gonads. Signs of ovarian damage exist. Nobody's studying it well.
I like it. And there is something to the idea of blood goes to the heart twice, but it goes on two different sides. And so we ought to be able to we ought to be able to look at the damage and actually read this pattern into it.
I don't buy this, precisely because we have a four-chambered heart, and there's a little bit of slosh, but for the most part, the four-chambered heart keeps the pulmonary and the systemic circuitry completely separate. And so the cardiac muscle, which is doing the pumping, has within it these chambers, these two atria and two ventricles, each of which gets, half is full of blood at any given time, but half of it just came back from the lungs and half of it just came back from the body, and never the twain shall meet. That's what four-chambered heart means. A three-chambered heart, there's a lot of mixing. A two-chambered heart as well. But yeah, we and birds and crocodilians, if memory serves, with our distinctly separate pulmonary and systemic systems, don't have the mixing. So
I don't understand what that falsifies.
Well, what is it going to a spot twice? If blood isn't going to any spot twice, it's going to you've got a vessel, and half of it is here and half of it is here. Why would it? I don't understand. Because
that's two chances. So first of all, I'm not compelled that I understand the circulation well enough to know that it would end up there first, but it gets two chances to lodge in the heart somewhere, right?
I guess so. It comes back through some part of the heart, and the idea. Maybe.
I mean, the thing is, you ought to be able to. I would love to know whether or not there is an asymmetry based.
So I thought it was about a double dose in a particular place, but no, okay.
Yeah, somewhere in the heart. Yeah. Yep. All right. Well, it's an interesting question.
Yep. Bret, according to the hypothesis you just laid out, why is myocarditis occurring more frequently in men?
Well,
it might in part be the catecholamine connection.
Could be. Let's put it to you this way. There is an asymmetry between male and female that is pretty much written into the universe. Interestingly, it re-evolves with multiple instances of male and female. That causes males to be stronger and more fragile. And so men live shorter lives, are stronger at their peak. And this has to do with the game theory surrounding sexual reproduction. And so it is quite likely that one of the advantages that men have in sport has to do with them being built for extreme performance in something like, let's say, warfare, right? Mm-hmm. And that extreme performance is part of a trade-off, and that it borrows from longevity and things like that, and that basically, that the heart is more capable and more fragile in men by design.
Yep. Which doesn't have anything to do with the hypothesis, but it doesn't need to. Like, a hypothesis to explain, to propose an explanation for the myocarditis that sometimes follows mRNA vaccination is not expected to, nor should it be expected to, explain all of the variation in myocarditis.
Right. And in fact, again, the subclinical manifestation is we don't actually know that this is more common in males. What we know is that it's diagnosed more frequently, and therefore it may be that females tend to have subclinical cases because of differences in heart design, etc.